You found the skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Alrighty, well, hello everyone. Welcome back. It's me, it's Jackson Wheat, and also one of my favorite guests is the Dapper Dinosaur.
Welcome. I'm so much of a favorite guest that I'm still not on the intro.
Dang.
Starting right off with a burn.
Oof. Retired, I guess.
Yeah, early retirement. Send condolences to Jackson.
To be fair, if you want to be on the intro, you might start complaining to me instead of to Jackson.
That's true. Peter is the one who oversees the technical aspects.
But then I can't make that joke every time.
Peter, do it.
That is true.
Kneecap him.
That is true.
All right. So we know you guys loved our last series so much. And actually, in terms of views for a live stream, it did very well on my channel.
I bring in the views.
That's true. You absolutely do. You probably passed me up in subscribers by now.
Have I?
I have no idea, but I assume so. At any rate, so Dapper he's got that top hat and the monocle, and everyone loves dinosaurs. So everyone likes looking fancy, and everyone likes dinosaurs. So what's not to love?
There's nothing not to love.
Exactly. So the last series that we did was on the process of and the evidence for evolution, and I think that went pretty well. Overall, we covered, most of the sort of bare bones of evolution. We didn't get into the what?
I was gonna say it was like evolutionary biology, like 101, crammed into like 8 hours?
I think it was 6 hours because it was
6 hours.
videos.
Okay.
Yeah.
Oh, okay.
So,
yeah, there you go.
Yeah. So the basics of evolution, an entire class just crammed into 6 hours. So you're welcome. You can save that money. Don't go to college. No need.
I disagree with this advice. You may want to go to college if you desire to be a professional in any field involving evolutionary biology.
Fair enough, I guess.
Technically, there are no degree requirements to submit and have papers published, but it helps.
That's true. Yeah, that is true. And also, if you are planning on submitting a paper, don't say that you got an okay from said paper, put their trademark on it, and then lie that happened.
Yeah, that's what we in the biz call academic fraud.
Yeah, and if you commit that, you pretty much have no standing in the academic community ever again at that point. So
even if you later do good work, no one cares. There's very little forgiveness in academia.
Yeah. Yep, exactly right.
But so today I think we're here to talk about the first 80% or so of Earth's history.
Yeah. So all right, here's the thing, everybody. We in the same way that we had slides for the evidence for evolution, we have made up a slideshow of the history of life on Earth and geology, etc. And we're calling this natural history. So we're basically going to cover about 80% or so of the Earth's history. We may get through all of the first PowerPoint today. It's not very long because neither of us are geologists, and so we're going to be focusing predominantly on the biota, which didn't even show up until about 500 to 700 million years after the Earth had coalesced. So there's a little bit of time there. And also a lot of chemistry involved in all this sort of stuff. And microbiology. Microbes are sort of the... they were in charge for most of Earth's history.
I mean, there's still the majority of the biomass on the planet.
And that's true. Yeah, I think the first macroscopic organisms appeared at the start of the Ediacaran, which was about 635 million years ago. So... And then you have the Cambrian explosion about 540 million years ago, which we'll get to, of course. So, yeah. And bear in mind, life started somewhere between 3.8 and 4 billion years ago. So from about 4 billion years to about 635 million years ago.
Yep. Which is
That
one of the
was
reasons
just
why
microbial life.
I hate it when people compare modern microbes to what they imagine the first life would have to be like. Because, like, look, man, by the time you get to anything that's recognizable as a modern group of microbes, you've already gone through, like 3 billion years of evolution.
Yeah.
A lot can change in that time.
Bent Hoven, no relation, asks, the Precambrian, is that before the Cambrian? And guess what? You're correct. The Precambrian, and then there's the post-Cambrian, which is not really a term anyone uses, but it's okay.
We could just start using it.
We could. I suppose we could. I don't...
Post-Cambrian is pretty much pre-flood.
Oh, goodness.
Oh, there it is. All right. Let's jump right in, Peter. The Precambrian. It's not even really fair to use this as a term because it encompasses multiple like eons, which are longer than the Phanerozoic. So it's just a useful term that which I think originated before anyone had any idea how long before the Cambrian was.
It's kind of like CE, right? The Common Era.
Right.
That's pretty short compared to the whole span of human history. Who cares? We're just going to go with it.
Yep. It's just in the same, just like species puts on heretic hat. Precambrian is just a useful construct. Takes it off. All right. Okay, getting spicy already. Let's go to the first one.
Is that still a spicy take?
No, probably not.
Okay.
But anyways. Okay, so as said, we're not geologists or I don't know, what would it be? Astronomers?
I'm not an astronomer or a geologist.
Cosmologists? Yeah.
I'm also not technically a biologist either, so
I guess also technically I am not yet. At least I would not consider myself one yet because I'm not actively contributing to the field, so I would not consider myself a biologist.
I'm a biology enthusiast.
There you go. That's a good point. So the theory, as near as I can understand it, currently is that our solar system, and that includes our Earth, coalesced from a pre-existing nebula about 4.6 billion years ago. Not much I can really say about that. That's what I understand. And the Earth had cooled enough because the Earth was very hot. When you have lots of these different bolides all smashing into each other, that creates heat. Lots and lots of heat.
Yes, it does. Just the initial collapse is going to cause a lot of heat because you're taking potential energy, converting it to kinetic energy as those things come towards the center. But then when they actually coalesce at the center, that kinetic energy then has to turn into thermal energy. So Earth basically starts well above the melting point for iron.
Right. Yeah, exactly. So very, very hot. And so, as you can see, it took another 200 million years for the Earth to have cooled enough for liquid water to exist on our planet. That's quite a while. Think about that. To put it in perspective, 200 million years ago was in the Jurassic, from today. So Stegosaurus, I don't even think Stegosaurus was around yet. He was like late Jurassic, right? So that was...
yeah, but I honestly don't know when the taxon arrives on the scene, like what year. Sorry.
Fair enough. But still, that's a long time. That's before flowers, before birds.
It's long before flowers. It's actually questionably before birds. It depends on what you count as a bird.
Okay,
because birds kind of arise in the mid-Jurassic, but are they crown birds? What does bird mean? Crown bird?
Okay. Yeah.
So there were flying feathery things, but they probably would not have been something that you would recognize as any modern bird group.
Right. Yeah. Suffice it to say, 200 million years is a long time.
Yes, it's much longer ago than, say, George Washington.
Only marginally.
A few orders of magnitude.
What's a few zeros between friends, right?
I mean, compared to some of the people we deal with, not much apparently.
Fair enough. And as the Earth was substantially warmer than it is now, it also makes sense that the composition of the atmosphere was different. But also, there weren't biological processes yet that were contributing to the composition of the atmosphere either. And we'll get into that, I think, in a few slides from now. But the point is essentially that the early Earth's atmosphere was composed of a number of compounds which exist in only very minute quantities in our current atmosphere due to a variety of processes. So that's hydrogen sulfide, methane, and carbon dioxide were the main ones. So the atmosphere was primarily reducing at the very beginning. It doesn't seem to have been the original hypotheses about the origin of life was that like the atmosphere was reducing when life came on the scene, and that was the Oparin. What do you call Oparin? Well, he was one of the guys.
Oparin Haldane?
I think it was Oparin Haldane. I think you're right.
Okay.
They proposed that back in the early 1900s. Darwin didn't really care about what was going on with abiogenesis. He was just kind of like, yeah, you probably had like, a warm pond and some phosphorus and salts and things like that, and that probably gave, was involved in the origin of life. But he wasn't super concerned with it. He really didn't write much of anything about it, and so it wasn't for another, he died in the 1880s, so it wasn't for another like 40 or so years before anyone was really sort of trying to figure out what the early Earth's atmosphere was like and how this would have contributed to the origin of life. And then using the Oparin Haldane hypothesis, you had the Miller-Urey experiment, which is very well known, which happened in, I believe, was the 50s, right?
I do believe that is correct, although I would, before you write this on a test at school or something, Google it. Don't take our word for it.
Yeah. So you had the Miller-Urey experiment, which generated a few amino acid compounds and some other organic molecules, and that was really neat. That is in no way, shape, or form the state of the field nowadays. So
no, in the 70 years since then, we've made some progress.
Right, and it's really funny because there is a certain guy going around who is claiming that that's it. Who is an organic chemist, a synthetic organic chemist, who is apparently claiming that that's the only experiment that's ever been done in the field. That was it 70 years ago. So that's a little weird. But anyways, as you can see, there was a lot of water vapor, and there was a lot more carbon dioxide than there is today. Carbon dioxide's it's like less than 1% of the atmosphere now, isn't it? Something like that.
Yes.
It's a very small percent.
If I remember correctly, it's currently measured in like parts per billion or something.
Yeah. So,
which also means, by the way, that any amount that you add is actually fairly significant because it's already such a low amount. Weird.
Strange.
Yeah.
Very weird. And so there was a lot more carbon dioxide in the atmosphere in the past, much more so than today. There was not a lot of free
parts
oxygen.
per million. Michael Apple corrected me on that when I was wrong about billion. Parts per million.
Thank you. Who all do we have in here? Hello, everybody.
Dessel Drace, Michael Apple, Kappa 1611. We got Smitty. We got Paleologos is here. We got Professor Flynn, Bent Hovend. Vandalia is here. I think that sums it up.
Yes. Welcome, everyone. Glad to see everybody. What did they say? What was the
carbon dioxide? It was a thing. Fortunately, we don't have as much of it now as there used to be.
Yeah. Was there land during the Hadean? I don't remember. I know
at first it would have been all land.
Right. But I mean, there was all land, and then when it allowed for when the Earth was cool enough for water to form, I believe it was all like one continuous ocean, and then you had the first tectonic plates arising later. But I don't remember. I think there's a PBS Eons video on that. So I'll give you a controversial paper that argues otherwise. This is just a chart. This is the chart that I found that is. Oh, well, thank you, Michael Apple, for for $5. Appreciate it. This is just the chart that you typically find associated with atmospheric change throughout Earth's history. Is this probably exactly correct? Probably not, but I think it suffices for our purposes.
Yeah.
So, at any rate, anything to add, Dapper?
No, I think we're pretty good. We'll probably, when we get later into this, talk more about the rise of oxygen during the Proterozoic.
Correct. Yeah. Okay. Next slide, pulls. All right. The origin of life. There's a book on the origin of life that I read a while back. It's titled "The Vital Questions" by Nick Lane. He's a biochemist. A really interesting book because the sort of I don't want to say dumbed down. It's the, I guess, the sort of,
it's the for lay audiences.
Yeah,
book.
for lay audience picture that you get is kind of the, like, the black smoker, which are the hydrothermal vents in the deep ocean, and it's like life probably originated around the hydrothermal vents. Well, there are different types of hydrothermal vents, which, I mean, yeah, makes sense. And the one that was originally thought to be the place where life possibly originated, they're called black smokers. They give off this deep colored smoke, which contains lots and lots of different minerals and molecules and all sorts of stuff in it. And the thinking was, well, maybe because it's giving off such a mixture, maybe this contributed to the origin of life. Well, maybe, but it seems that researchers have kind of moved away from that and more towards what are called alkaline hydrothermal vents. So rather than giving off these big combinations of molecules, they're giving off this very basic water. Alkaline refers to the second column of elements, right?
Well, so it has to do with what kinds of ions you're giving off in solution with
Right.
water. Whether you're donating protons or receiving them. But there's the alkali metals, but there are things that are alkaline that aren't the alkali metals.
Okay, gotcha. But in these vents, you have, so the water coming out of these vents is like full of hydroxides, which is very basic, or I guess it would be hydroxyls.
It's almost as basic as Jackson wheat. Almost as basic.
Slightly more acidic, right? And as these are being given off, the thinking is that since there was a greater amount of carbon dioxide in the atmosphere, this would have dissolved at greater rates into the ocean, which also today impacts us in terms of ocean acidification. It still, still dissolves in the ocean and contributes to the decalcification of corals and sponges and mollusks and things like that, which is not great. Not great at all, especially if you rely on some of these organisms for food.
Imagine
really
being unable to grow your skeleton because there just isn't enough calcium that you can absorb.
Yeah.
You might get this thing. I think it's called rickets. It's really, really bad.
Yeah. Slug rickets or snail rickets.
I mean, I doubt that the symptoms are exactly the same, but I imagine they're similarly severe.
Yeah, no, absolutely. Yeah. Because with corals, when they're under like thermal and acidity stress, they bleach. They essentially expel their algae, and they die. And that's not great. That's not great, especially when fish, which are fished for food, tend to live in coral reefs or around coral reefs, or at least spend part of their life cycle in coral reefs. So then that's really not great.
Correct.
But so the idea is you have these natural proton gradients. You have minerals which catalyze the reaction between these between the like carbon dioxide in that's dissolved in the ocean and the hydroxyls which are coming out of these vents, and you can catalyze the formation of organic molecules. And so the simplest organic molecule is methane, right? CH4, isn't it? It's just one carbon with 4 hydrogens.
I am not a chemist. I was very bad at chemistry in school.
Well, you also get your carbohydrates are CHO.
It's like it's in the name.
Yeah. No, we're not going to tell the story. Anyways, it's too dumb. So, or I believe it's CH2O. Sorry, not CHO. But then you can, you can add as many carbons and hydrogens and oxygen as you want and generate these really, really long carbohydrates, which have different functions and all sorts of stuff. So basically, all of our necessary organic molecules are found like in and around these vents, and the thinking is that maybe this contributed in some way to the origin of life, which entirely possible. As I said, not a chemist, but that doesn't seem super wild to me. The fact that we are able to so easily find organic molecules in space seems that the production of organic molecules is relatively easy. I would be willing to bet, though, that the actual origin of life itself is probably relatively difficult to accomplish. But at its core, life is really just about, maintaining thermodynamic equilibrium with their environment?
Disequilibrium.
Disequilibrium, sorry, yeah. Thermodynamic disequilibrium with your environment. And that's
basically what it is to be alive, is you are maintaining a thermodynamic disequilibrium by using energy gradients in your environment to conduct like biological processes. When that breaks down and you can no longer maintain thermodynamic disequilibrium, there is no more energy gradient for you to exploit. You have reached thermal equilibrium with your environment. Therefore, you cannot conduct any of the life processes that require energy. And so you're not alive, because that's what life basically is it's the system that conducts these processes. Once it can't do them, that's it. So if you want to get super reductive about it, life is just a self-sustaining series of chemical reactions that maintain an energy gradient with the environment.
Mm-hmm. And Dessel Drace points out also, along with cobalt, tungsten, nickel, and several others. Yeah, exactly right. So, yeah, lots of different metals involved. Yep, yeah, you are correct, Dessel, but as said, neither of us are chemists, so... But we appreciate the input. All right, next slide, please. And here's just a series of different reactions that that go from like it says inorganic molecules to organic precursors to your building blocks and functional polymers, and ultimately to earliest life, because it would have been a gradient, right? None of this was you had some inorganic molecules and then boom, bacteria. That didn't happen. You would have had a series of steps, probably occurring through a variety of processes, which ultimately ended up at what we would consider living, right? And at what point do you consider like replicating molecules living, right? There's a lot of debate over that, because you also have things like viruses, for instance, which replicate when inside a host. But are they living? Well, I mean, they do lifelike things under certain circumstances, but not under others. And what about like satellite DNA, DNA which just replicates on its own, or prions, which are just proteins? There's not even any DNA involved. At what point do we consider things living? And the argument or the response to that is sometimes, oh, well, if they live independently of something else. Well, the problem with that is there are bacteria that are totally dependent on host cells, like chlamydia, for instance. So That's not really a good response.
Which infect a ridiculous proportion of koalas.
True. Very true.
Koalas got the clap, man.
Skip entirely in a publication or spend 2 hours trying to trace every pathway. Well, it's on the internet. So where can you find this chart? I don't remember where this particular chart came from. If you search maybe abiogenesis chemical pathways or something to that effect, you probably be able to find it. I don't quite remember where this came from, but at any rate, next slide, please. And then, same sort of process going from atoms, molecules, macromolecules, polymers, protocells to full cells. And you have these processes are occurring along inorganic surfaces at first, and which ultimately would have been replaced with organic membranes. So just kind of reinforcing that. Next slide, please.
Jackson, as the producer, if Paleologos wants to have that chart and you have that as a picture, I'm more than happy to be a real producer.
Yeah, no, you can send it to him
and make sure that he gets it. So.
Yeah, absolutely. I don't remember where I got it. I apologize, but yeah, feel free to hand it out to whoever wants it. No problem.
Okay.
I don't copyright or own any of these things. These are just pictures I got from the internet. Some of them come from technical papers. Some of them I just went to Google Images and they work well enough. So, Luca was the last universal common ancestor. So the first cell and Luca were not the same thing. Probably. Very likely.
Yeah, probably not.
Luca refers... Go ahead, Dapper.
I was going to say, there are actually hypotheses for the first cellular life either post-dating or predating Luca.
Very interesting.
Yeah,
but
I tend to think that predate seems pretty likely, since all extant life has pretty similar cellular membranes.
Right.
But I mean, I'm willing to entertain the other option that more than one line of life developed. What we think of as cellularity after Luca.
Yeah, I know because that works for some things, like the transcription enzymes evolved differently in bacteria and archaea, meaning ultimately eukarya also.
Mm-hmm.
So yeah, so some things evolved in separately in both, because there are ideas like that Luca was a ribosome genome, or had a ribosome genome rather than a DNA genome, and so that quote the enzymes evolved thereafter. It's also likely a population where members were exchanging genetic material amongst themselves rather than one individual, which is more the case when we get to like common ancestry between like animals or plants or fungi, something like that. So at this point, probably not one individual. Or possibly not.
Yeah, I doubt that it would be one individual.
Next slide, please. Okay, so prokaryotic world. So life, the first life would have been prokaryotic in the sense of it just didn't have a nucleus. But Dapper, entertain them for a moment, please.
Well, everybody, so yeah, like Jackson was saying, for the first 4 to 2 billion years ago, life was what we would call prokaryotic. Prokaryotic is sort of a grade, and it's not like it's a clade, because eukaryotes nest within a clade that also includes prokaryotes. But basically, they didn't have a nucleus. When we say it was almost entirely unicellular, we do get some colonial things that may have had some kind of division of labor among cells, which would make them sort of in the in-between stage between unicellular and multicellular, but we're still primarily talking about either independent living single cells or colonies of single cells. So Stromatolites, which, by the way, you can still see being formed today in Australia, are some of the earliest fossils that we get, and we get them going back billions of years. And these are columns of photosynthetic algae that are not, well, bacteria, which is a kind. These are a kind of algae, whatever, that sort of grow as time goes on. So you get a layer of this bacterial film, and then as it reproduces, the newer bacteria start going up because that's the direction they can grow. The lower down bacteria end up dying, and the intercellular matrix that they were exuding to help stay in place kind of solidifies and becomes part of the rock, basically. And so this is some of our earliest evidence of life at all. But also there are possible bacterial fossils. Like we have this one in the lower left hand that's about 3.5 billion years old. Now, is it definitely a bacterial fossil? No.
I'm glad you pointed that out, actually.
Yeah, but it is.
There's a bit of debate over that.
It's consistent with what we might expect for a string of bacteria, because some bacteria form these filaments where they just connect end to end, and they do it for various reasons. A lot of photosynthetic bacteria will do that. It helps them, keep their place in the water column and things like that. And they can also share resources that way. So Brainbug asks, were these phototrophic? So as far as the potential bacteria on the left, who knows? Maybe. But as far as stromatolites, yes.
Yeah, the cool thing about that, so I put possible there because William Schopf, I think is how you pronounce it, it's like SCHOPF, was the one who found, and a team found those possible bacterial fossils. And I mean, I have no problem with them being bacterial fossils. That would certainly fit in with like the known evolutionary history of this group.
Yeah, it doesn't break anything for them to be for that to be a fossil. It's just the data doesn't quite support that as a firm conclusion. Yeah,
it could be some abiotic, like minerals or something like that, which just happen to sort of look like it. Because you have to remember, these are microscopic. I mean, look at that. It says 10 micrometers.
Yeah, these are very, very tiny.
Very, very small. You're not seeing these with the naked eye. They're probably, poring over these rocks under a big microscope, and they happen to see that and be like, "Oh, what's that?" But yeah, and also almost entirely unicellular. Yeah, you're right. Like, what does it mean to even be multicellular? Because cyanobacteria like Nostoc and Anabaena, they do things that look sort of multicellular-y.
I go with cell differentiation on the basis of division of labor. So if you have more than one, basically, like expression of the genotype of the same, more than one expression of the same genotype in a collection of cells, and those differences in expression are the result of adaptations to different function, especially with reproduction being a single function set aside to a particular group of cells, that's when I say that it has gone from being colonial to multicellular. For me, that's what I use. I think that means that all the things that everyone agrees are multicellular, they all fit that bill. And then the stuff that everyone agrees isn't multicellular doesn't. And so I think it's a fair definition. But hey, you know what? I don't get to decide that, but I don't know. It's what I go with.
I mean, I didn't agree with that. I mean, because, you have guys like Anabaena where they have a cell which just fixes nitrogen while everyone else is still doing photosynthesis, right? And it's like, okay, well, this is a line of clones, but one of them is doing something that the rest of them aren't doing. Yeah. So is that multicellularity? Well, maybe.
I'm going to go, if that's a maybe, but once they have a group of cells that are just there to reproduce, then it's a yes.
Yeah. Yeah, and same, I think, for actinomyces, which were originally accidentally misidentified as fungi, hence actinomyces, that refers to their fungi, that refers to fungi. And so, but they're a group of bacteria. They're not fungi at all, but they kind of look like, I think it's mycelia. And so there's a little bit of confusion over that. So, it also makes sense that there would be this gradient of organisms. Some are definitely unicellular, and some are definitely multicellular. And then if there were these transitions from one to the other, and in fact there are transitions in both directions, funny enough.
Yep. It's almost like what you might expect if evolution were true.
Right. Yeah, exactly. This is what you would expect if evolution were true. Yeah. Things that are sort of kind of halfway, partway between two different things. So anyway, anything else? Are we ready?
I think we're ready.
All right, next slide, please. Just more on the last universal common ancestor. This was a recent paper. I don't know what I was thinking, because with the evolution PowerPoint, and I put my sources with everything. I don't know why I screwed up so badly on this one. My apologies, audience. But this was a recent paper where they were arguing you can basically divide bacteria into 3 groups. And so you got, that's right, yeah. So Gracillocutus, and Terrabacteria, and Fusobacteria, which is basically derived with respect to both. Terrabacteria includes the Firmicutes and Cyanobacteria. So for reference, Firmicutes are the gram-positive bacteria. Well, for reference, gram-positive. Yeah. Well, yeah, a bunch of them. So for reference, if you take a microbiology class, for instance, you're going to come across these terms that are gram-positive and gram-negative. So most bacteria are gram-negative, and some of them are gram-positive. Now that refers to their membranes. So gram-negatives typically have like this one membrane, and then gram-positives have these big double membranes, right? Isn't that it? Oh no, they have a lot of peptidoglycan in their.
That's actually something you should ask Beethoven about. He knows more about that than I do.
Yeah, I believe they have a lot of not their membrane, their cell wall. Sorry, my bad. I meant cell wall, not cell membrane. And the cell wall is composed of peptidoglycan, and so when you are doing your gram stain, they turn different colors. You use these different stains, and you can be like, okay, well, this one's purple, and then if it's gram positive, and this one's like red if it's gram negative, because they don't absorb the stain that you're putting on them, and so you can differentiate them based on that first, and then you can subject them to all sorts of tests about do they make a particular enzyme? Do they have a particular metabolic pathway? All that sorts of fun stuff. So anyway, horizontal gene transfer is common among bacteria, and so that's kind of why it's difficult to make a single phylogeny of bacteria, because you can have this thing called, what is it, the hologenome, I believe it is, where it's like you have what the bacteria in this, what you can find in the bacteria in this group, but that's not all the genes the bacteria in this group may have, right? And the reason is that they get genes from other bacteria, or even from viruses, or they pick up from the environment, things like that.
Bacteria are great at horizontal gene transfer.
They are. They do a whole lot of it. Yeah. Both absorb the stain. Gram-positive just don't easily get the stain washed out. That's right. Correct. Let me put that up here. According to KH says both absorb the stain. Gram-positive just don't easily get the stain washed out during the ethanol step because of the cell wall. Exactly.
The ethanol step is my favorite step.
Yeah. Black Queen hypothesis. I'm not sure what that is. I've heard of the Red Queen hypothesis, but that's a new one on me. Okay. Anything to add, Dapper?
No.
Okay. Next slide.
I'm sorry. You did a good job covering that, Jackson.
Oh, thanks. So kind. Okay. So I know thus far we haven't really talked about. We've gone now about 2 billion years into the evolution of life on Earth, and we haven't even touched on archaea. Sorry, we'll get to them in a moment. So, so the Great Oxygenation Event. So this is when we start to see appreciable levels of oxygen in the ocean, and then which outgassed to the atmosphere. Now, not nearly as much as there is in the atmosphere today. We're talking going from parts per million to greater than
20-ish%.
yeah, it's we're not going all the way. As you guys saw in that chart at the beginning, it was like a slow rise at first, and then it really went up later. And that really big sort of pulse in oxygen didn't occur until, I believe, like around the Cambrian, which has also contributed to some hypotheses about predation and raising the size limit of organisms, which, again, we'll get to that later, probably next week. So the ancestors of cyanobacteria utilized electrons taken from reduced manganese, and they oxidized the manganese to perform photosynthesis. Other bacteria already do this with iron, hydrogen sulfide, and organic carbon. And so the evidence of this is found in oxidized manganese deposits dating to 2.4 billion years ago. So while we don't have a fossil record of photosynthesis, because this is a metabolic pathway and thus very, very unlikely to be fossilized, researchers can make predictions about what should be found if this evolved from a prior metabolic pathway. And indeed, the fact that there is oxidized manganese before a jump in oxygen indicates that the bacteria, probably the ancestors of cyanobacteria, were using this, and then they went a few more steps and developed that photosynthetic pathway, and then we're really cranking out oxygen, right? And so by 2.1 billion years ago, cyanobacteria had evolved oxygenic photosynthesis, pumping oxygen into the atmosphere, and the evidence of this is from what are called banded iron formations, and there's a little picture of one down there on the bottom. So these formations have iron in them, and they rusted, because that's what happens when you get oxygen and iron in a room together. You get rust.
Right, because it's iron oxide.
Yep. Anything you'd like to add?
Oxidation happens to your fruit, but that doesn't mean you can't eat it anymore. It just looks a little different. So when you get all those brown spots on apples or bananas, that's okay. That's apples after you cut them, If there's a big brown mushy spot, then maybe it's not okay. But, just saying, oxidation, it's a thing. It's fine. Don't worry about it.
Dessel Drace says, I guess referring to the Black Queen hypothesis, it's an attempt to is that supposed to be examine trends of reductive evolution of bacteria. Lane gives some alternative answers in his book, Power, Sex, and Suicide. Well, I'll add that to the list, I guess.
Yeah, you're reading recommendations.
One more book to add to the Amazon wish list. Thank you, Dessel Drace. Okay. Alrighty. Next slide, please. Oh, also, Benthoven says, I trust me infinitely more times than Uncle Kent. No relation. Fair enough, I guess.
That's very fair, yeah.
So here you go. You have the great oxygenation event, so you get a big jump. And look at that. Yeah, we're not even up to present atmospheric level, so you get a really big jump around 2.1 or so billion years ago, but it's still not up to today in terms of oxygen. Then you get, it says overshooting, so oxygen levels actually went back down apparently, and then they sort of stabilized for a while, a period known as the boring billion, which we'll talk about momentarily. And then you get another raise right around the start of the Cambrian, and then it sort of goes up to modern levels from there. So, just sort of an overview of that process. Anything you'd like to add?
We may get into it later, but at first, this oxidation was not great for life, because oxygen is actually pretty bad for life overall. The only reason that it's so important for, say, aerobic organisms, like me and you and whatnot, is because there have been some heavy adaptations to allow life to use it to basically more efficiently break down some molecules to get the energy. but it's still very damaging to things like DNA and all sorts of other biomolecules who will break down when they're oxidized. So, when oxygen first came around, it was extremely toxic to basically everything.
Yeah, and you still there are still lots of things that incorporate oxygen which harm your cells, like peroxide, hydroxyl radicals, superoxide, all that fun stuff, which can damage your cells and your DNA, and so you have to have peroxisomes come around and sort of nip those in the bud.
Yeah,
and DNA repair mechanisms and all kinds of stuff. So yeah,
also,
it's still toxic, even though.
I was gonna say that's why ozone therapy is one of the stupidest things imaginable.
What is? Oh god, I'm afraid to ask. What is ozone therapy?
Oh, it's people doing things like huffing ozone or dissolving ozone into a liquid and then taking it intravenously. It's extremely dangerous. We've
been around for far too long. I'm convinced.
Yeah. So what? This is a bit of a tangent, but what ozone likes to do is it just likes to rip apart carbon bonds. Any carbon bonds around? Well, not after ozone. Guess what? A lot of you is made of.
Is it carbon bonds?
Yeah, it's carbon bonds. So,
oh my goodness,
ozone in high concentrations basically just dissolves the molecules making up your cells and turns them into a soup. If you ever want to see what it does to you, you can take some ozone and spray it on a latex glove, because latex is also mostly carbon bonds. It just frays apart into nothing.
That's what we call a yikes, maybe a yowza.
And yet it's being promoted by, medical woo practitioners as the next greatest thing, because it's going to attack all the bad stuff in your body, which is true.
I mean, that is true.
It's just that it will attack the rest of you too. It's just like, hey, in a laboratory setting, flamethrowers have been known to kill cancer. Correct.
They're not wrong, I guess. If you eat those what is it, like the cyanide pills that Hovind was talking. Yeah, you'll never have any problems again, I guarantee.
Yeah, take enough of them, you definitely won't have cancer.
Oh gosh. Okay. Anyways, next slide, please.
Oh, by the way, I just did a video about Kent Hovind where he said, imagine putting something toxic inside of your body while talking about vaccines. This from the man who was like, just eat a bunch of cyanide, it'll be fine.
Oh no.
And also the man who famously said that vitamins is an abbreviation of vital minerals.
Oh, goodness. Oh, man. Is there anything he won't screw up?
No.
We're not here to talk about him. Uncle Kent can wait until...
I was a high school teacher for 15 years.
No.
Anyways, the origin of eukarya. So we have promptly dashed through about 2 billion years of the history of life on Earth. Sorry about that, folks.
That's all right.
And here we are. Somewhere between, I think it's like 1.8 and 2 billion years ago is when the origin of eukaryotes is hypothesized to have occurred. And yeah, I said popular. It is probably the most well evidenced hypothesis. That's why it's the most popular. That's how things tend to work in science. Things gain popularity when they are evidenced. That's how that works. As opposed to social media. Anyway, so the idea broadly is that an Archaean, whom we haven't really talked about, Archaea is the other megaclade of life. Basically, you have extant life. You have bacteria, and then Archaea, and eukarya branches within Archaea. And I'll actually show a phylogeny for that in like the next slide or the next something like that. Which was also a surprise. Because for it was thought, essentially, Archaea were discovered to be separate from bacteria in the 80s, I think. And then once they were discovered to be something different, it always seemed like Archaea were closer to eukaryotes. But it wasn't until later that it turned out eukaryotes are actually a subset of Archaea. They're Archaeans that engulfed a bacterium which seems to be either in or near the alpha proteobacteria, which is one of the phyla of bacteria. which is one phylum of bacteria. And so that alpha proteobacterium, it is thought evolved into our mitochondria. And so this relationship drove the evolution of the cell nucleus, other organelles, and possibly even sexual reproduction. And another endosymbiotic event occurred in the ancestor of plants, Archaeplastida, so the red algae, green algae, all those guys, which became was a cyanobacteria, and then that second endosymbiotic event, it gave rise to all the photosynthetic red algae and green algae, and then some green algae and red algae were then taken in by other eukaryotes, and then you have like endosymbioses within endosymbioses within endosymbioses. It gets really wild because protists are like, hey, can I use this to make food? Absolutely. I think I will.
Mm-hmm.
So you get like these little Matryoshka dolls, and some of them, very interestingly, still have the remnants of the nucleus within their degenerate green or red algae. It's called a nucleomorph, if I remember correctly. And organelles don't really need a nucleus, so that seems pretty good evidence that this was another endosymbiotic event. Also, there are things like the fact that the chloroplast in plants and mitochondria in all eukaryotes have like a double membrane, and that is or the reason for that is the inner membrane is the mitochondrial, or is the bacterial membrane, and the outer membrane is one that came from the eukaryote later. Anything you'd like to add?
No, I think you covered it very well.
Okay. Alrighty. Okay, next slide, please. So here we go. Here's a phylogeny of, you got Archaea. So you have bacteria way down there. They're the outgroup. And then from within the Archaeans, eukarya branches out. And in recent years, there is this group of Archaeans that has been discovered called the Loki Archaeota, which come from an area up near, I think it's like Iceland.
They fight the Thor Archaeota.
I think Thor Archaeota is actually one of the genera. It's like, I think it's Thor, Loki, Odin, and Heimdall. And each one's like Thor Archaeota, Loki Archaeota, etc. They went all out.
I approve.
And so they appear to be the closest prokaryotic relatives of eukarya. They have some enzymes and other components which are more similar to eukaryotes than to other Archaeans. So, all right, next slide, please. And then here it is, more fleshed out. And then, feel free to read that. So you have your Archaean, which is that guy with the it's got like the blue and the gray, and then the red there is the pre-mitochondrian in a sense. And again, we're not chemists. I'm not an organic chemist, and so I'm not really going to go into all the details on that. But anything to add, Dapper?
Nothing related to this, but apparently at one point someone reported me for changing the colors of my avatar to the colors of the Ukraine flag because they thought I was implying that Ukraine was going to be extinct because my avatar is a dinosaur.
Uh-oh.
So
the Asgard Archaea, that's what it was. Sorry, not the Loki Archaea.
Sorry, I just got that news and it was a little, I was like, what? Okay. Yeah, actually, Jackson, you know far more about this section than I do. I my knowledge base gets smaller and smaller the farther away from eukaryota you go.
Well, you're in luck because next slide, please.
Yay!
Da 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 da. We hit eukarya. For any Archaea fans out there, I know we just kind of brushed over them. Big sad. Archaea Archaea are weird. Lots of them are like thermophiles and halophiles, and they live in some really weird places.
Mm-hmm.
Just generally being weird, or maybe we're the weird ones, because here we are with nuclei, and
I mean, look, I think I made this point before, but like prokaryotes form the majority of the biomass of the planet. Like,
so we're the weird ones.
Not only are eukaryotes outnumbered by prokaryotes, they're just outmassed. If you put all eukaryotes on one side of a scale and all prokaryotes on the other side of a scale, the prokaryotes would go down and the eukaryotes would go up. So like eukaryotes, especially multicellular macroscopic eukaryotes, are like a vanishingly small portion of the biosphere when you take a look at the whole thing. It's just that because we're in that section and we interact with it, it's the part that we tend to pay the most attention to.
Yes. Square root of 2 points out that eukarya are actually included inside the Asgard Archaeota as of now. So that's cool. So yes. So it can't. So can it even really be called? I guess if Well, I mean, I guess it can if all the members of Asgard Archaeota are like monophyletic. They're and like sister to eukarya. Then I guess it would still be fine. But I don't know what the precise makeup of that clade is right now, and I don't think the researchers really do either. I mean, this group only came out like a few years ago, so this is very cutting edge, or it was very cutting edge of research. So anyway, there are lots of tiny things that are out there in the oceans, and we have not found very many of them, comparatively speaking. So eukarya.
Yay.
Agree with Dapper. Yeah, eukaryotes happen to know a little bit more about. So in a broad sense, you could sort of classify eukarya into, we'll say, 4 clades, 4 big clades. And I'm saying this pedagogically because it's wrong. But in a broad sense, you can sort of classify eukaryotes into 4 major clades. One is your excavata. Sorry, I was drawing a blank there for a second. Even though it's on the slide, I see that now.
It's got a star.
It's got a star. One group is your excavata. One group is called uniconta, and I put that in quotes, or amorphia. One is what is called chromista, or we'll just go with chromista. And one is archaeplastida. So those are essentially your 4 major groups. Now, I say essentially because excavata is probably not monophyletic. It is probably para, if not polyphyletic. And this tree that you're seeing right here is not the most up-to-date tree in the world. The problem with that is if I put any tree up here representing broad eukaryote phylogeny, it's going to be different tomorrow.
Yeah.
Sorry.
Yeah, this is... The evolutionary tree of life is the most uncertain at the very base and the very tips. In the middle, there's actually pretty we've got a pretty good handle on a lot of that stuff right in the middle. It's really not shifting around very much. But once you get so basal that you're doing a lot of stuff like, very long ago evolution and a lot of horizontal gene transfer and stuff, it gets complicated. And then at the very distal ends, you're starting to do stuff like, are these various things like they might be convergent, they might not. It gets hard to tell at those very, very distal tips. And so here we're at the sort of basal most part of the eukaryote, phylogeny. So yeah, it's a little. That's where it moves around, is at the bases and the tips.
Right, exactly. And there is a lot of this moving. I think, for instance, like Cryptista and Haptista are included among the chromists nowadays. So Cryptista isn't considered closer to archaeplastids, for instance. And I believe Hemimastigophora nests like sister to Archaeplastida. And who knows where the excavata end up. Ancera monadita, that's another, that's like, maybe it's near Amorphia.
What we got to do is we just got to get DNA sequences, full genome sequences for all extant organisms, and that'll help clear it up.
Well, yes and no, because you can also have like fast evolving regions.
Yeah, and you can have horizontal gene transfer.
I mean, like, in some cases, absolutely, having whole genome sequences can help. But also having more data isn't always necessarily the thing that's going to solve it, right?
Mm-hmm.
So I mean, it depends. It's like a case by case basis. But broadly speaking, yeah, we want more genomes, which gives us more stuff to compare. So, because more data does definitely help over less data. So, you guys don't really need to worry about most of these groups. We're not really going to stay on them for too long. Excavata, that's like where you have Euglena and some other weird little guys. Funny enough, I think it was Metamonada was thought to be like a pre-mitochondria eukaryote.
Didn't it later turn out that it seems like they lost the mitochondria?
Well, they have sort of neo-functionalized them. They don't look like mitochondria anymore. They're called like, what is it, mitosomes, I think. And there's one other group that happened to, where it was thought that they didn't have the mitochondria, but they've just been refunctionalized. Although, fun fact also, now in recent times, it has been found out that there are some excavata that don't have mitochondria. So they still lost their mitochondria anyways. Just, additionally. So fun stuff. There's like there's an Idarian that was also discovered, which doesn't appear to have a mitochondrial genome. It's a member of the clade Myxozoa, which are these predominantly fish parasites within Nidaria. So,
at one point, they were thought to be single-celled non-animals. They're really, really, really degenerate in the strict sense, where they have lost most of the things that you would think of as what animals need to have.
Mm-hmm.
But after their genome was sequenced, it was like, oh no, these are jellyfish. Like, oh, okay, sure.
Paleologos points out you need all the data before coming to any conclusions. But as I said, well, I mean, having more data is good, but having a genome doesn't always necessarily resolve everything, because as pointed out.
Well, I think he's got the winky face because you can never have all of the data. There's always more work to do, but you have to come to conclusions.
I guess that's true. That's true. So I don't think we really are going to talk about the Chromis too much after this. So they're the SAR group, the Haptista and the Cryptista. Although I think they're really fascinating groups. That's where you have your kelp, diatoms, foraminifera, radiolarians, coccolithophores. Those are all within the Chromist group.
It's the chalk group.
Yeah, it's the chalk group. Yeah, got lots of cool, a very wide diversity of protists. Also stuff like
Jackson, come on, protists. That's not a real thing.
Lots of cool little single and multicellular eukaryotes distributed across a wide variety of ecologies. Anyway, all right.
Hey, when I was in school, Protista was still a thing.
I kind of want to complain because I took a class in college, a zoology class, where the professor mentioned there were 5 kingdoms, and I about had a stroke.
Oof.
So
anyway, that's rough.
Next slide. Origin of multicellularity. So as that picture shows, it occurred a lot. Yeah, there we go. The bacteria. There are some groups of bacteria with cellular division, but they're generally considered unicellular. So cyanobacteria and actinomyces. Since eukaryotes have or are making lots of ATP, which is adenosine triphosphate, that is the essentially the currency of molecular energy for your cells, because they are chugging out way more of this than your average bacterium is capable of doing. This allows for them to have organelles, even though there's some debate about that over certain groups of bacteria, but broadly speaking, you can have a larger genome and organelles and cytoskeleton, all kinds of cool stuff, because you have more energy, right? You're getting more energy, and this allows you to do more, and that makes sense, right?
I mean, it's actually a basic thing about like economics, which is it basically works for anything where your resources are limited. So there's some kind of scarcity. It happens in human societies where people specialize for different jobs. It happens in eusocial organisms like ants and bees, where you have different or individual organisms specializing for different tasks that the whole colony needs. And ultimately, it's the same thing that's happening in multicellularity, where you have different cells that are specializing to work at different things, and so they become more efficient at that one thing, and then the entire group gets to benefit from that specialization and that division of labor.
Right. Yeah. Absolutely. So yeah, simple and complex multicellularity has evolved over 20 times in eukaryotes. So green algae, red algae, brown algae. Brown algae is like the kelp. Those guys, Macrocystis, Laminaria, all those dudes. Fungi, animals, etc.
Now,
So,
Jackson, is this tree rooted or unrooted?
This is unrooted.
Okay. I know that there's been some confusion about that with some people, so I just wanted to make sure that we're clear about that.
These cowards should have just rooted it. What are they doing?
How dare they?
Because it's just a big polytomy where you have excavata down on the bottom.
Actually, I only see one polytomy.
I mean the tree itself, because they're all coming out of the same.
We might have to have a discussion about whether or not that counts as a polytomy later.
It's not saying any is more closely related to each other, as I can tell. I mean, it doesn't look like it.
Doesn't matter.
Okay. Anyways, so yeah, multicellularity occurred lots of times in organisms, in different groups of organisms. Probably, within fungi, it probably evolved multiple times. It evolved within chromista multiple times. It evolved within like green algae multiple times. So, and it's probably in response to different pressures. It's probably not always the same pressure. When we talked about the Saccharomyces experiment in our evolution PowerPoint, we discussed how there was selection for gravity. We talked about for the ability to settle out faster as a result of gravity. With the Chlamydomonas one, we talked about predation. And predation, it probably fueled at least some of these. I wouldn't be surprised. But there are lots of different environments that organisms can inhabit, and so predation was probably not the only selector. It's probably a variety of things. Anything you'd like to add?
Just that is one of those nodes on the right labeled other?
Other straminopiles.
Oh, okay, there we go. Got it.
Diatoms. All other
I was just like, what is other for? Okay, no, I got it now. Yeah, I'm just not looking at that. Okay, good. I was very confused for a second.
All right. Next slide, please. Yes, the boring billion. So eukaryotes evolved somewhere between 1.8 and 2 billion years ago. Thereabouts. That's when we have the earliest eukaryotic or probable eukaryotic fossils, which are called acritarchs. Although I remember now I added a slide about that. So we'll get to that later. Nice multicellularity chart. Can you give me a citation? I like I said, I don't know what I was doing. Why I didn't include citations. Just look up on Google Images evolution of multicellularity, and I can almost guarantee that chart will come up with the associated paper. I do apologize. I'm normally better about this. Don't know why I didn't include citations for these pictures like I did with the other PowerPoint. My bad.
Very disappointed, Jackson.
I know. Just take me out back already. So the boring billion. So about 1.8 billion years ago, you have the supercontinent of Nuna, which lasted from 1.8 to 1.3 billion years ago, and then Rodinia from about 1.3 to 0.75 billion years ago. And so you have erosion and breakup of Nuna led to trace elements ending up in the oceans, potentially lifting restraints on the evolution of novel features. That was from a paper I read, which I cannot remember the author, but I believe it was a recent paper. And then I say split about this time. They split the estimates from what I've seen are about 1.6 billion years ago or so, or 1.6 to 1.8 billion years ago is when Amoebozoa is thought to have split from Opisthocanta, according to some molecular clocks. So boring refers to, and this was a term that came up that was invented by paleontologist Martin Brazier. I think it's like Brasier or Brassier maybe.
Was he French?
I don't think so. That's
No. And you probably have to pronounce all the letters. French is that language where it's like about a third of the letters are probably just not going to get pronounced.
Yeah. Dapper, we're gonna. I have to read this comment. Dapper every stream. We're gonna have a little talk later, Mr. Jackson. Yes, dear. You're not wrong. You're not wrong.
And we usually do have a little talk afterwards.
We do. We have a good chat. We always have fun. So boring refers to the fact that the chemical content of the ocean and the atmosphere were relatively stable for this time. So, which is it's funny because while the geochemistry was relatively stable, life was undergoing a lot of changes. So whatever erosion was happening was allowing these trace metals to enter the ocean, and these could have been incorporated into the various organisms that were alive. So evolution was definitely happening. And if you look at molecular clocks, a variety of different like the major groups of organisms were splitting at this time period, in this time period. So you have from the origin of eukaryotes at the beginning to like the origin of animals down towards the end of the boring billion. So lots of evolution occurred. It was not boring for organisms by any stretch.
It was just boring for rocks.
Which isn't it always boring for rocks, basically?
I mean, look, I understand that there are people who are apparently very enthusiastic about rocks per se.
Mm-hmm.
And you know what? I'm not going to kink shame.
Fair enough. Fair enough. Gotta get your rocks off, I guess.
I think that's what erosion is.
Oops.
Literally, you're getting the rocks off of whatever, right? That's erosion, isn't it?
I guess you're right.
Yeah.
Yeah.
See, it all comes back full circle.
Next slide, please.
And that means that volcanism is getting your rocks on.
SR and seawater. That's strontium, isn't it? SR. So I think that's strontium. So apparently strontium was doing its own thing during this time period.
Good to know.
Cannot be contained, become ungovernable, as it were. So this chart is backwards. Most of our other charts, they typically start at whether your start time is at the y-axis and then going outwards. But this is you're getting closer to the y-axis, is coming closer to today.
Yeah, I don't like it, guys.
I'm not a big fan of it either.
When you're putting time on the x-axis, the past goes to the left.
Yeah. Yeah, not a huge fan of this chart, but at any rate. So there you go. Yeah, the boring billion, you can see from about 1.8 to 0.8 billion years ago. And there's some wiggle room in there.
There's a major lubrication event. Jackson, I hope you flag this as not appropriate for children.
I mean, I believe I did.
Okay, good.
Oh, this was all intentional, by the way, folks. This was all planned in advance. No. So look, so we do have rising continents there somewhere about 2.5 billion years.
Oh, there you go. I like how it's like gig a year. That's a fun. It's a fun unit right there. Gig a year.
Gig a year.
That's what it is. It says age. GYR. That's gig a year.
You're correct. That's what that is.
That's a good unit. I should start measuring things in gig years. Like, I'll be there in about 14 times 10 to the negative 8th gig years, give or take.
Relaxed Bayesian molecular clocks for the win. Yep. Squishy lid. Yep. Squishy lid. All rock stuff. Lubrication, squishy lids. All very important. Apparently the rocks were not bored, despite what Martin Brazier said.
I mean, they're having squishy lids in major lubrication events, so no, apparently not.
And also and
new plates.
Snowball Earth.
Oh, and Snowball, yeah.
And Snowball Earth, which we will, I believe there is a slide about that shortly. So next slide, please. Oh, Acritarchs. Yeah. So Acritarchs are the remains of organisms that have yet to be assigned to any particular clade. So technically, Acritarchs could be bacteria or protists or even animals or plants.
Well, they can't be protists because that's not a thing.
Wow.
Yeah, I'm calling you out on that one, man.
Good lord. But they could fall into any of those real or fake groupings of organisms. Because that Acritarch is just a waste basket. Anything that we don't know yet what it is goes in there, but if you figure out what it is, it goes into whatever clade it should
was definitely alive. We just don't really know how.
Yeah. And so some of them, like Dictyosphaera, Valkyria, and Tapania, are almost certainly eukaryotes. So they display degrees of cellular complexity that is comparable to eukaryotes, and so they were probably eukaryotes. And so the oldest eukaryotic acritarchs come from that formation, which I am not even going to attempt to pronounce, but dates about 1.7 billion years old. So yeah. Anything you want to add?
No, I think you covered that pretty well.
Okay. Next slide, please.
I have to say my patented line for these shows.
Fair and balanced, copyright, trademark, restricted. So we get to our glaciation. So interestingly, prior to, I think it was like within the past 20 years, the Cryogenian, which is named for its snowball or slush ball Earth periods, didn't actually start with those snowball and or slush ball Earths. It started before that, and there was a geologists kind of got together and were like, "Hey, since the Cryogenian is named for these events, we should probably make it start at the first one." And so they did.
Imagine that.
They were like,
way to try and make sense, geologists.
Yeah, I mean that, I appreciate it. I approve. Because what is it? A lot of times, when you get into the Phanerozoic, which we will next time, periods tend, but not always, to start and stop with major extinction events.
Yeah,
it's totally arbitrary where you start a period or end it, but it's a convenient metric to have it start and end at different major events, right?
Well, because there's noticeable differences in the rocks, right? Whether it be the paleobiota that you find, or whether it be the actual physical nature of the rocks involved, and things like that. So, like for the Cryogenian, what's distinctive about it? Well, in terms of geology, you get a lot of things like noticeable, like glacial tills, drop stones from glacial deposits, stuff like that in the rocks from this period, because that's what glaciation events do. So there's this period of definitive glaciation. So that's a period,
right? Yeah, exactly.
Same thing with the Mesozoic. It's bookended by gigantic extinction events.
Right, and typically, and the end of the Triassic is also, and the start of the Jurassic is also marked by a major extinction event. But interestingly, the Jurassic Cretaceous kind of isn't.
No, it's just sort of like it was a really long span of time, so break it up sometime. Put a line there.
There were smaller extinction events, and of course, we'll talk about those way later. But yeah, in
a few billion years, we'll talk about those.
Well, at this point, it's a couple 100 million years.
Oh, that's a good point.
But yeah, we'll get there in a couple 100 million years. So stay tuned.
So like 0.2 gigayears. It's my new favorite unit of time is the gigayear.
So these, so if I remember correctly, what triggered these snowball Earth events is you basically have all of your land masses in these supercontinents. Now, a supercontinent doesn't necessarily mean all of your land masses are together, because the Europe, Asia, and Africa also form a supercontinent because they're all connected. But obviously, not all of the continents are part of that. However, with Rodinia, that wasn't the case. It was this one big continuous landmass. And it ended up sort of at the equatorial regions, and this resulted in lots of erosion, which an erosion sucks CO2, carbon dioxide, out of the atmosphere. And remember, carbon dioxide is a greenhouse gas, so it has a tendency to trap heat in the atmosphere, which, is a good thing. We got to have some heat on this planet, or else we just freeze. But
like all things, it's a moderation thing.
Right, exactly. And so as this carbon dioxide is being majorly sucked out of the atmosphere, this is causing cooling. So lots and lots of it is exiting the atmosphere, and this is dropping the temperature of the Earth, and this is what caused the snowball or slush ball, because I think more recent research has shown that it probably wasn't totally iced over. Probably equatorial regions had open water. So, but still really cold. Pretty darn cold.
Much colder than the last glacial maximum.
Yes, yeah, absolutely. And animals probably evolved during or just before the start of this period, somewhere between maybe 800 million years, 720 million years ago.
Go animals.
Yeah, which were probably like sponges.
Yeah, the animals at this time would not have been much that you would recognize as an actual animal. Like, probably none of them really were motile as adults, and they were all probably like suspension feeding and whatnot.
Right. Yeah, exactly. All right, next slide, please.
But they had cholesterol.
That's true. They did have those steranes, which I think I have a slide where I talk about that. If not, we definitely will bring it up.
Nice.
I'm
seeing some Kimberella, some Dickinsonia, some.
I've come around on that point.
Oh,
yeah.
have you?
So the Ediacaran. Yeah. Oh, well, again, we'll explain it in a minute. So the Ediacaran. This is the final period of the Neoproterozoic before we get to the Phanerozoic, which is the Cambrian to today. So the Ediacaran lasted from 635 to 542 million years ago, and it is in this period that we see the first macroscopic life. There's lots of microscopic fossils prior, but the first macroscopic organisms appear here. And at the beginning, you get what's called the Avalon explosion, which is where you see those weird little frond-like guys, which you can see in the back of this picture. They're red, things like Charnia. So these weird little frond guys, which I believe are, based on recent analyses, considered to be like stem eumetazoans. So that's interesting.
Yeah, we know that they're not photosynthetic because they're generally found in deep water deposits where there wouldn't have been light. So despite looking like a fern branch or something, they weren't photosynthesizing. So currently we're thinking that maybe they were filtering particles out of the water, or maybe they were just passively absorbing various minerals and whatnot that were dissolved in the water and then using those to power a very slow metabolism.
Right. Yeah, and there are some guys out there, some like actual legitimate paleontologists. It's a very, very small group, though, like Greg Ritalik.
It's so small we can name him.
Like, he's still putting out papers, that get cited. Not as much, I think, anymore, but he's still out there doing stuff. He's still, excavating and whatnot. Who claims that a lot of the Ediacaran deposits are, in fact, like terrestrial deposits. And no, just no, they're not.
They're not. Mm-mm.
I think whatever sedimentological arguments he first sort of offered have been kind of debunked, and he's still sort of pushing it for some things. And it's like, no, they were marine. These were marine depositional environments, and these are marine organisms.
Yeah.
So, kind of, it's sort of. I don't think he's quite. Well, maybe he is. I don't know. I'm not.
I mean, we're finding them in things like shale and stuff. Like these aren't being found in like alien sandstone or something.
Right. I found a fossil sea pen before. Very nice. That's
nice.
Really cool. There was actually a thinking. Paleologo said that there was thinking briefly that Charnia was in fact a sea pen or some other Nidarian, but that turned out to be false because it's a fractal. It is not built like sea pens are, or any Nidarian or any organism alive today, really.
Yeah, we don't really have fractal animals anymore. There's an argument for plants being fractal, like some of them at least.
Okay.
Like to some extent, because branching patterns are well approximated by various fractal patterns mathematically, but not animals. So, whereas we get a lot of fractal growth in the Precambrian, where we get this branches that have branches that have branches, all in things that are apparently animals. You also get what's called glide symmetry. I don't know if you're going to talk about that.
No, go ahead.
So glide symmetry it's sort of like having segmentation, but instead of having a segment that goes across the body, laterally, each quote unquote segment, it's called an isomer, is actually sort of shifted up. So if you look at, I don't know, do you have a better picture of like the Dickinsonia there?
Yeah, a little bit later. Oh, well,
Okay.
if you want to bring this up later, I guess you can.
Yeah, so we'll talk more about glide symmetry when we get a better picture of Dickinsonia, because Dickinsonia is an animal that displays it pretty well. And I say animal because I do believe that was actually one of the taxa for which like molecular remnants of cholesterols were discovered.
That's one of them. Yeah.
Which makes it a very definitive animal.
Yes. Yeah.
Because plants don't have cholesterol.
Right, and not just any cholesterol, but like animal-specific cholesterols.
Yeah.
So there are like you'll find like your aberrant protists that may make like some kind of cholesterol or something.
Except
but, like,
you won't, because those don't exist. Jackson, you're going to have to stop saying protist, man, or I'm going to bring it up every time.
There was also an increase in oxygen levels towards the end of this period, which is implicated in some of the hypotheses about like the evolutionary arms races in this time period, because this is when you also first see the evidence of predation. You find like boreholes in Cloudina shells and things like that. So somebody was getting some Cloudina meat.
Yum.
Just digging on in there. Tasty stuff. Which are also apparently annelids now.
Oh.
Which kind of cool.
Interesting. I did not know that.
Yeah, paper came out, I think, on that last year. I could be wrong, but I believe that came out.
And Tully Monstrum is apparently a vertebrate.
I've heard that. I mean, I guess I wouldn't be surprised, but it's a weird dude. Anyway,
I was holding out for mollusk, but whatever.
Yeah. Well, we all face disappointments in life, my dinosaur friend. Next slide, please.
Tully Monstrum not being a mollusk is just one of them.
Precambrian biota. So here are some non-animals. You got Orosphera duraldiae, which is a fungus, the earliest fossil fungus dated to 1 billion years ago. Bangiomorpha, a red algae. Now I put Bangiomorpha here, but actually there is an older red algae that has been discovered from 1.6 billion years ago, and it's called Raphatasmia. I believe it was discovered in India.
Nice.
But yeah, again, these guys are microscopic. This says, what does that say? 25 micrometers, I think. Very tiny.
Yeah, that does say 25 micrometers.
So yeah, real small guys. And then testate amoeba, 742 million years ago. So that's Tony, and that's even prior to the Cryogenian period. So yeah, which also makes sense given what the molecular clocks we talked about earlier. Then Megasphaera is also technically an acritarch because it's a multicellular eukaryote, but isn't assigned to any particular clade. And this is from I'm drawing a blank. I can't remember the name of the biota, but it was discovered back in 98, I believe. And this is one of those fossils where the researchers were like, I think this is an animal embryo. And some of the alleged embryos from this fossil site are probably animal embryos because they apparently exhibit enough diversity in their cell types that this is only like comparable to animals. So yeah. Next slide,
there
please.
You go.
There you go. There you have it, folks. Archaeplastida. Okay, so here we are talking about plants again. So Archaeplastida is broken into 3 groups. You have Viridiplantae, which are the green algae. Rhodophyta. Rhodo means red, so the red algae. And then Glaucophyta. We don't really talk about the Glaucophyta. They're called the blue-green algae or green-blue algae, whatever. But so few people even know what those are. It's not even really worth discussing. So
I petition to change the common name to the GRU algae. GRU is a technical term, Jackson.
Okay.
It is. It's not biology yet.
I believe it. So again, the secondary endosymbiotic event, which the eukaryote incorporated a cyanobacterium, which became the chloroplast, that occurred prior to the evolution of these guys. And then you get your split, your red algae. If you like sushi, you eat red algae. That's called porphyra, which makes the wrap.
It's delicious.
Yeah, pretty good stuff. There are lots of different types of red algae. What do you call them? Urchins like eating red algae, as well as kelp, which is a brown algae, so very distantly related. And then the green algae are I believe next slide, please. I believe I go into more detail on them. Yes. Well, sort of. So this is Chlamydomonas. The little picture is a microscope photo down on the bottom left. So Chlamydomonas is sort of like what you could say the quote ancestor of plants would have looked like. He's just a little green guy with 2 flagella. And so their life cycle is pretty simple. He exhibits what, as we discussed in the evolution PowerPoint, a zygotic life cycle or a haplontic life cycle. So in essence, you are haploid for the vast majority of your life, even if that isn't a very long life, developmentally speaking. You are the only time you are diploid is when you're a zygote, when 2 gametes come together. It's not even really like which is male, which is female at this point. It's just which is the opposite. 2
of them.
Yeah, which is the opposite type.
Yeah,
mating type.
It's called a mating type. Yeah. So there you go. Sorry.
Oh yeah, no, and we don't talk about fungi really, but fungi can have like hundreds of mating types.
Yeah. It turns out sexual reproduction is way more complicated than male and female.
Yeah.
And there's a whole lot of variation within the umbrella of sexual reproduction.
Yep. Yeah, absolutely. So this is probably what the ancestor of plants would have been doing, just a little biflagellate photosynthesizing and reproducing every so often. But as we showed in the evolution PowerPoint, they can actually be induced through successive generations to become multicellular. So that's kind of cool under certain predatory pressures. Anything you'd like to add?
Well, now that I'm unmuted, I think you did a very good job covering that.
Oh, thanks.
But also, your slide does say a flagellated protist, which is impossible.
Next slide, please. Okay, so within Viridae Plantae, so we were looking at Chlamydomonas. So Chlamydomonas is unicellular, and from there you can, stack. If you just stack cells on top of each other, you get a filament of cells, or in the case of Volvox, you turn them into a little sphere. All right, make a little jelly that kind of holds everybody together, and you become a little sphere of essentially Chlamydomonas altogether. This is Proterocladus, this guy on the top right, which is a little filamentous algae from the Precambrian. And there are still lots of filamentous algae out there today. That's Spirogyra down there on the bottom right, called Spirogyra because here's the thing: the chloroplasts form a spiral. Crazy world. Yeah.
Is it a spiral or is it a helix?
Yes, absolutely. Next slide. Oh, there it is. Yeah, on the bottom. So you can stack your little Chlamydomonas on top of each other and get a filament, or you can put them together and make a little a sphere, and you get like a Volvox. And so all those guys you see on the bottom, Chlamydomonas, Gonium, Euterina, Pleuterina, Volvox, those are all alive today. Now, that's not a true phylogeny of them right there, because some of those are more closely related to each other than others. You may get like maybe Gonium is like more closely related to Volvox or something like that than to the others. So that's not a phylogeny. That's just showing in terms of what exists today, what the possible steps could have been. You also get this cool thing as you get like more complex, you add more cells and do more like division of labor. You go from being isogamous, where your gametes are virtually identical, towards anisogamy, where you start to have differences between them. Your egg cells get larger, for instance, than your sperm cells, to oogamy, where you have very, very different gametes. That's what animals are. We're oogametic because the egg cells are not motile, whereas and they're way, way bigger than our sperm cells. So And then this phylogeny of Viridiplantae. So you see land plants, quote, quote, that's the technical name for that is embryophyta, because they form an embryo. Although there are some other streptophytes which are closely related to them called the charophytes, and some of them also do similar things. So basically, the big difference in embryophytes from the others is these other guys release their gametes into the water, basically, and they come together. Whereas your land plants, you have your actually, I think it's on the next slide. I believe. Could we go to the next slide, please? I think I explained this there. No, I didn't. Oh, well, I don't know what I was doing. We'll get back to the reproduction thing in a second. Another way you could do it is you just make a whole sheet of cells, and that's basically what ulva is. So ulva is also multicellular, right? But its gametes still look like little chlamydomonas. So you have your haploid gametes up there in the top right, which is infused to form a zygote, which then grows into a sporophyte. So that is your diploid form, which then makes spores. It undergoes meiosis to form haploid spores, and then those haploid spores undergo mitosis to form gametophytes. So a sporophyte makes spores, and a gametophyte makes gametes. This is true not just for ulva, but also for the embryophytes. The same thing is true of your mosses and your ferns and flowers. It's all the same. It's just you're changing how long each of these processes occurs, basically. Anything you'd like to add on this point?
No, I think you covered it quite well.
Okay, next slide, please. In essence, basically, there are lots of different ways of going multicellular for plants. You can be like just a little blob of cells, or you can be a filament, or you can be a sheet. But basically, all you're doing is you're just like stacking your cells. Essentially, you're connecting them in some very simple way. So these guys are not like, it's not super complex. It's just your filaments. I mean, they're certainly more complex than like chlamydomonas is, but it's just a matter of you're not inventing the wheel, not reinventing the wheel. You are changing what already exists for new purposes. And that was something we tried to impart in our evolution slideshow, was that you don't have to make new genes for everything. You can keep your genes, you may duplicate them and then mutate those, but it's homologs of the same genes.
Mm-hmm.
So,
also, horsetails are cool.
Indeed, horsetails are cool.
They used to be much more prominent during the Mesozoic, and they were much, much bigger in some cases. Not in all cases. Just saying, because you got one out there.
Benthoven says, I used to have fewer cells.
That's true. Yeah.
And then one day you will have fewer cells again. So,
yeah. Take
pride in that fact, I guess.
Well, I feel like the number of cells in an adult human tends to vary through time.
Wait, did you say a horsetail was up here?
Yeah, is that not?
No, that's not a horsetail. That's Kara. That's an algae. I'm sorry, I wasn't.
Darn it.
I was reading the.
Looks like a horsetail.
Kind of, yeah. I'm sorry, I was reading the comments. I was sorry, my bad.
I could have said anything.
Yeah,
you would just been like, yeah, sure, Dapper.
It took me a moment. I know I've become Joe Rogan, basically.
I could have been like, oh yeah, protists, it's totally a thing.
Like, I've heard this thing said, and
snails
people are saying
ecdysozoans.
And you're telling me it's true, so it must be. I'm not saying it is true. I'm saying people should do their own research.
Yeah, I'm just asking questions.
No shade thrown, of course. Anyway,
no, some shade thrown. I'm okay with throwing some shade.
So the cool thing is, Kara represents a very significant step towards the embryophytes, the land plants, and that very significant step is your embryo develops on you rather than somewhere else. So instead of releasing your gametes to the ocean or river or what have you, you're keeping them on you. And so you get fertilized, basically, and then you can form, so you have these the oogonium, and then that forms your zygote, and then you release your zygote, basically, instead of just releasing the gametes. And so we'll get into more of this a little bit later once we finally meet the land plants. But you can see their reproductive strategy was predicated on these algae, which are definitely not land plants. So like I said, it's steps towards complexity. It's not all in one go. So you make your antheridium, which has your sperm cells, your oogonium, which makes your egg cells, and then the egg cell gets fertilized, and then that forms your zygote. So yeah, and then your zygote gets released. Next slide, please. Unless you have something to add, Depper.
No, I think you covered that quite well.
Okay, holozoa.
Yay!
So we are bouncing all the way back from plants and algae to much closer to animals now.
Yeah.
So we kind of skipped amoebozoans. Sorry, amoebozoa fans out there. Apologies.
Wait, you mean we didn't evolve from amoebas?
We evolved from amoeba-esque organisms,
but not actual amoebas.
Not members of the clade amoebozoa. No. Sorry.
Take note, everybody.
We just evolved from pond scum. Goo to zoo to you.
At least it rhymes. I like it.
It does rhyme. Gotta give him that. Look at this. I cited a source for once. Go Jax.
Well done.
I know. I appreciate it. So holozoa is the clade, Opisthocanta is the clade that includes fungi, ichthyosporia, philisteria, coniflagellata, and metazoa. That's the animals. And within that clade, holozoa is basically all those guys minus fungi.
Get out of here, mushrooms.
Exactly.
And
your favorite, yeast.
Cell wall. Well, yeah. So that is my favorite fungi or fungus. Yeah. Saccharomyces cerevisiae. What?
This little guy right here.
There's nothing funny about Saccharomyces. Jackson will just laugh at anything I say as long as I say it in the right tone. Is basically what happens.
That's not incorrect.
Yeah.
I think I laughed at the drinking rules like every time you said them. So
every time so far, yeah.
I still think tells a joke that doesn't land is the funniest one.
Well, it's because it makes it land for us.
No, wait. There's also what's the other one? Tells a joke that doesn't land, and that definitely didn't happen.
Oh yeah, stories that didn't happen. By the way, in case anyone isn't familiar, these are the drinking rules that I have for my Tuesday shows, where myself and a guest talk about a creationist video where the creationist involved has some kind of pun relationship with their name to my guest's name. So with Jackson Wheat, we talk about Dr. Charles Jackson. And with Ben Hoven here, we talk about Kent Hoven. And we also do an Eric Hoven with Erica Gutsa-Gibbon, and a Matt with Maddie. And there's two Matts. So we were on Matt Powell first, and then we switched over to Raw Matt. I think Maddie drew the short stick on that one. Sorry, Maddie.
Oh, Lord.
Yeah, that's...
that poll.
You get Charles Jackson. That's leagues above either Matt.
Oh, God, yeah, absolutely.
And it's still really bad, even with Charles Jackson. So anyway, that's enough shilling for my channel. Let's talk about ichthyosporia or something.
Yes. Holozoa, so this clade, and this little guy, Bicellum brazieri from Scotland, is apparently the oldest known holozoan, so dates to 1 billion years ago. So that's pretty cool.
A whole gig a year.
That is 1 gig a year, yes. Next slide, please. Okay, I guess we'll just, yeah. Okay, so Ichthyosporia, Philisteria, not a super lot to say about them. Ichthyosporia includes like some fish parasites, hence Ichthyo. They also used to go by this name called drips, which referred to, it was like an acronym for the members I can't remember their. I think R was Rosette Agent, which was like they diagnosed it before they knew what it was, or like had a genus or whatever. So it was what caused this. So so it used to be called drips or Mesomycetozoa is the other name for it, but Ichthyospora sounds cooler in my opinion. And then Philisteria. So both of these clades have members who can like do or no, actually I think they're both are they both unicellular? Or does either of them do multicellularity? I don't think so. I think they're both unicellular.
I'm pretty sure everything on this in this section is unicellular except for animals and some fungi.
I think you're right. So yeah, and then
some of them are colonial though.
Yes, yeah.
Like a lot of choanoflagellates are colonial.
Yeah, we get to choanozoa. So a choana is these are like a collar. So you have these villi, and they form a little collar around a central flagellum. And if I remember correctly, there's not really a huge difference between villi and flagellum, right?
The villi are tiny.
I mean, yeah, but it's like the same structure, isn't it?
Yeah, as far as I know, it's very similar. Yeah, they're. I mean, it's like, when does the cilia become big enough to stop being a cilium? I don't know.
Right. Sorry, I should have said cilia, not villi. My bad.
Yeah, villi are what you got in your lungs.
Yep, I'm sorry. Cilia. Sorry, not villi. Yeah, I don't think there's much of a difference in terms of like a microstructure between cilia and flagella. I don't think. If I'm wrong, someone point it out in the chat, please.
Yes, Yellow Jackson.
Please do. I love it. So there's so interestingly, like sponges, for instance, have what are called choanocytes, which are these cells that look a lot like choanoflagellates. They have sort of the same, or a lot of the same microstructure. Are they convergently evolved, or are these the remnants of choanoflagellates? Well, there's a bit of a debate, but I mean, I really wouldn't be surprised if it turns out they are, in fact, sort of homologs of choanoflagellates.
It's one of those things where like it could go either way, but it looks suspiciously similar, so there's a fair chance that there's homology there.
Yeah. This other dang, and I forgot to do it for this paper. So this picture comes from a recent paper, which I think came out last year, which is about how the choanoflagellates have their vesicular system, which kind of is or centralizes at their poles basically, and that's sort of similar to how like nerve cells work and things like that. And so maybe have this method of having your secretory vesicles at like the poles of a cell, maybe that was also accepted, and that came to be used for like synapses. And yeah, maybe it did. It's entirely possible. All it takes, and so as you can see in this picture, it's like you have your colonial cells coming together, and well, we already know that choanoflagellates will change their like orientation as a group in response to certain stimuli. So it's not crazy, I think. That sounds like a reasonable hypothesis to me, but not a choanoflagellate expert. That would be Nicole King. So if you have not read any of her papers, go do that. Next slide, please. I think we're pretty close to the end, so we can probably finish this up. Eosciathospongia, maybe a sponge. It's definitely sponge grade, but it's like I think the early Ediacaran, something like that. Hautia, definitely a Nidarian. There's been like zero argument that Hautia is a Nidarian. I think it's like even a crown Nidarian, which is kind of interesting. Then Dickinsonia, so a stem bilaterian. And we were talking about glide symmetry earlier, so feel free.
Yes. So the thing with glide symmetry, and you can actually see it here if you look very closely at this Dickinsonia picture, it has these little things that look like segments on either side. But if you look closely, they don't actually line up one-to-one. each one is sort of at the medialmost point is right sandwiched between two other isomers, and that's what that glide symmetry is like. So it's like it's been shifted, sort of like bricks, right? If you have like if you look at bricks on a wall and you look at just two rows of bricks, each one is usually offset by like half a distance of a brick. And that's similar to how glide symmetry is working. And so in a lot of these Precambrian animals, we're seeing glide symmetry. You also see it in Charnia. And that is distinctly different from the true bilateral symmetry that we get with things like Icaria and you or me, where both sides are actually directly mirrored. And it's not clear if one of those is ancestral to the other or if they were independently evolved or what. It's still sort of up in the air. It's, for one thing, we don't know the genetic basis for this kind of glide symmetry with isomers because we don't have any extant isomerically symmetrical or glide symmetrical animals. And so all we have to go on is the fossils, and that's Precambrian fossils are already hard enough to come by. So,
absolutely correct. That other little guy down there, who's I think only like a couple centimeters long, something like, or a couple millimeters long. I think.
Yeah, I was saying centimeters is pretty generous.
Yeah, no, I think it's only a couple millimeters. That's Icaria Wariutia. So a little bilaterian. So in the Ediacaran, that's where all these guys are from. You have like your earliest the earliest probable evidence of animals, it comes from steranes in the Cryogenian. So there's been a bit of an argument because there are these rocks from Oman which have these steranes in them, which are known from two groups of organisms. One is sponges and the other is like in very minute quantities from a type of algae. And and so these researchers who were looking at these rocks and doing these geochemical analyses, I figured we were pretty close to the end. They were doing these geochemical analyses. They found that there were large quantities of these steranes. And so what that seemed to indicate to them was that there were sponges here. And the reason they think it was the sponges, not the pelagophyte algae, is that the algae give off these sterane compounds as an accidental byproduct of a different metabolic pathway. And so you'd expect very low quantities of it if that were the case. But in fact, the quantities of the steranes match that for sponges rather than for the algae, because the researchers tested that, because that's what you do. You rule out hypotheses that could potentially kneecap your argument. And there's been a lot of back and forth over this. There was argument that maybe these like bacteria associated with sponges make it, but turns out they don't have any of the genes involved in that metabolic process. Then there was an argument that there are some groups of chromists who make like the precursors of these molecules, but a precursor is not the same thing as the actual molecules. So that argument has kind of gone by the wayside. And one guy tried making the argument that you could synthesize these or you could generate these compounds via abiotic conditions. However, the conditions he used are not found like on Earth or in the ocean. So, not super relevant. But anyways, so
they were abiotic elements brought to Earth by aliens.
Sure,
so they were like
possibly
abiotic, but the aliens brought them here just to trick people billions of years later. They were trickster aliens. Yeah, maybe.
Why not? So I tend to agree that probably the steranes are indicative of sponge activity, which again,
or sponge inactivity, since they don't do much.
But I mean, since animals probably originated about 800 million years ago, again, that doesn't fall outside of the timeline. Since it was like 700 million years ago, that still falls within the probable range of animals. So, okay. Next slide, please. A couple more. So now we're actually into the crown bilaterians. So you have Kimberella, which is a Lophotrochozoan. I didn't put mollusk because there is some debate over that. Is it a mollusk? Is it a stem mollusk? Is it a stem Lophotrochozoan? Well, it's what happens when you're dealing with organisms that are millimeters across.
It certainly is what you would expect. Something that's like karyotypic of the common ancestor of mollusks to look like.
Sure. Yeah.
It has sort of the most generalized mollusk type body that you can imagine.
Yeah. It was also like a soft, sort of biomineralized little shell on its back rather than a tough, like calcium shell or something. So, yeah, it some kind of Lophotrochozoan, maybe a stem mollusk, but Namacalathus.
Certainly not a stem bird, though. Definitely not a stem bird.
Probably not. Yeah.
Unlike pterosaurs.
I put some. Ooh, Namacalathus is probably a stem Lophophorate. So Lophophorata is the clade that includes brachiopods, Foraminida, and Bryozoa. And so this little guy seems to have some of the characteristics that they have. So this pushes their clade back into the late Ediacaran. Claudina seems to be an annelid based on some recent analyses of like possible preserved gut. So that's kind of cool. And Eulingia, well, I put panarthropod, but Eulingia could also just be an annelid, although it has these little trilobed segments, which are kind of like trilobites. So that's neat, which is why it is maybe panarthropod affinity. But the A in abiotic stands for aliens. Yes, it does. Alien biotic.
It specifically was the aliens from the Star Trek The Next Generation episode, The Chase. Fun fact.
I'm glad we know which ones it was.
Oh, yeah.
Next slide, please.
I mean, they left us a little like holographic video message in the DNA of numerous species across the galaxy. So
nice. Very nice. I knew it wasn't all junk DNA.
Yeah, some of it's part of a videotape.
Yeah.
Oh, okay.
And I mean, we're at the 2 hour mark anyway. So also, yeah. So this is from Evans et al. 2021. Yay! I got 2. Good job, Jackson. Proud of you. High five to yourself. So these are all Ediacaran fossils. You got Tribrachidium, which is this weird sort of triradial guy who seems to be sort of like a basal eumetazoan or a stem eumetazoan. Dickinsonia is like a stem bilaterian. Icaria is a bilaterian. And then Kimberella is a Lophotrochozoan. So you can see the sort of stepwise acquisition of characters in these different clades, or these different fossils, indicating that they are members of different clades. So you have a diversity of organisms that is starting to appear before the Cambrian. Hoo-hoo. Which is like exactly what you expect under evolution. Crazy stuff. I don't know.
I mean, it's certainly crazy that we even found these things, because you remember that it was not that long ago that the Cambrian had the earliest known fossils.
Yes. Yeah, I think it was only the 1960s was when researchers were starting to be like, oh, hey, these guys are from before the Cambrian. These fossils date to the pre-Cambrian. And yeah, so that was pretty recent. When Darwin was around, he thought the Silurian was the lowest layer.
Yeah.
The geologic column. So, that was 160 years ago. So, in a very short time, we've come pretty far in terms of our understanding of the history of Earth. And we still got a long way to go.
Oh, yeah. We're going to make all sorts of new discoveries.
Yep.
Thanks for watching. But before you go, I just want to say a special thank you to my channel members and my patrons, especially those pledging $20 or above. Work in Progress, Benthoven, Cynically Skeptic, Denny5252, Ian Chen, John Ackerman, Chris Love, Landon Knoll, Yepetus, Mabbity Babbity, Sphincter of Doom, The Venerable Bede, and Patrick Dennis. As you probably know, YouTube is a very volatile platform, and from month to month my income on the channel can swing wildly. But the people you see on screen are directly supporting so that I don't have to worry too much about that, and the channel can keep going as it is, and perhaps even improving. If you'd like to join this team, there are links below to join the channel or in the description to join the Patreon. The Patreon starts as low as $1 a month, and the channel memberships start as low as $1.99 a month. On Patreon, you can even get a discount for pledging annually. If you do decide to pledge, you'll get access to an exclusive Discord server just for channel supporters, as well as early access to all of my pre-recorded videos, often up to 2 months in advance. Higher tiers will unlock even better perks. Now, if an annual or monthly pledge isn't right for you, but you still want to support the channel, there is a merch store down in the description, as well as an Amazon wishlist, just for me. And if financial support isn't something you can or want to do, then if you still want to help out the channel, please just like and share these videos and make sure you comment on them. It really helps the channel out. Thanks again for watching.